All right. Well, thank you for the intro. Yeah, it's yeah. Can you hear me? Okay. Very good. Well, it's nice to be here. Thanks, Matt, and to the lab for the invite. Matt was at OSU just a few months ago. That's where we connected and got to talk a little bit. And his lab is doing cool stuff. And I've never been here. I've told him and Caitlin as well that this is the first time actually after almost 20 years in this, inside, not just as a tourist walking around on campus or whatever. But so it's nice. Yes. My lab is interested in bacterial cooperation and communication, and we are using model systems or the classical model system approach of how and why bacteria communicate and cooperate. We are interested in two foci. We have quorum sensing, cell-cell communication in bacteria. We focus on acyl homoserine lactone quorum sensing in Pseudomonas aeruginosa. This is the acyl homoserine lactone molecule, and the traits that are regulated, such as proteolysis and skim milk plates. And this just kind of looks nice for show and tell. There's many other traits that are regulated by quorum sensing in this organism. And then the other system we're interested in is iron acquisition via siderophores. Those are diffusible iron scavenging molecules that are expressed under iron starvation conditions. They look like this. They have sort of this yellowish green hue when the bacteria are grown under iron starvation. This is what the molecule itself looks like. It's a non-ribosomal peptide that chelates iron with high affinity. Sort of, I coincidentally gave a talk at our seminar series at OSU a few months ago, and so that was kind of primed me to look back a little bit, for almost 20 years, what have we been up to and how have we approached things, perhaps? Is there a common theme? And I kind of translated that over for today. I believe it's kind of the testing of long-standing hypotheses, sometimes perhaps in creative ways, as we like to think. Typically, as ambitious scientists, we like to see through the entire scientific method. We do and make an observation. We form a hypothesis. We devise an experiment, and then we iterate kind of through this process. We're never really done, right? But in our case, I think we take hypotheses that have been out there for a while and then we tinker a little bit and come up with experiments of how to test them. And really, most of this is on quorum sensing, what I'm going to tell you about today. And it perhaps looks a little bit like a laundry list, but I think it follows, nicely. One thing kind of fits into the other. And so I'll tell you about some evidence that quorum sensing is a social behavior that's subject to conflict. But before this, it's been looked at from kind of a mechanistic point of view, which is what microbiologists do most of the time. That social conflict within quorum sensing causes what we might call a tragedy of the commons. That quorum sensing and its sociality aspect is an intriguing antivirulence drug target. That quorum sensing is also a bistable switch, more mechanistic stuff now, that quorum sensing can even short circuit within the cell and not do what it's supposed to do, namely sense, population density. And then I might switch to siderophores and tell you about the costs that are associated with it, and then the propensity for cheating in that system. Okay. So I'd like to start out broad, perhaps appropriate for the audience, that there's a world of cooperation out there in animals, of course, that we are most familiar with. The eusocial insects cooperate a great deal. There are some spiders that cooperate as well to make these webs. And of course, these meerkat colonies, they have an intriguing division of labor with, for example, sentinels that, warn the colony of danger. And there is cooperation, however, at the microbial scale as well. There is cells of Pseudomonas bacteria that come together to form biofilms. There are other types of bacteria, for example, the marine bacterium Vibrio fischeri that associates with squid host to produce light when it is at high density as a camouflage mechanism, and also produces light when it is not with its host, but just growing in dense colonies on the plate. This is a paradigm for quorum sensing and symbiotic interactions as well. And then we have these curious bacteria. They're actually not fungi, they're bacteria called myxobacteria that form these nicely differentiated fruiting bodies, okay, with stalks and spores. The fundamental property that we can derive from all of these behaviors is behavioral cooperation is a shared investment in a group resource, okay, that they all have in common. And that comes in the form of public goods, typically in microbes. So public goods are more concretely things, products that bacteria produce inside their cell, but then release to the extracellular environment. And so they benefit the entire local community population. So types of public goods that I've already mentioned briefly, and others are proteases that have a role in extracellular protein digestion, for nutrient acquisition, siderophores that scavenge iron, antibiotics involved in microbial warfare. We have exopolysaccharides, the glue that keeps biofilm communities together. And then, of course, rubiophysceri that produces light as well. So there's a tension here that these public goods have an individual cost, but they have a group benefit. So they are in principle exploitable, and therefore we must explain the evolution and maintenance. And I'd like to cast this mechanism called quorum sensing in that light. But quorum sensing also would be a way that could perhaps restrict cheating by restricting the activation and the production of public goods when they're most beneficial, so they are least wasteful. And that would be at high cell density. That's when public goods are most beneficial. And this quorum sensing is then a mechanism that makes that happen, that restricts activation of public goods to high cell density by secreting these small molecule autoinducers, okay, that act as a proxy for population density. So let's consider a low cell density state where there is an individual bacterium that secretes these quorum sensing molecules. In case of gram-negative bacteria, these are acyl homoserine lactones. A generic molecule looks like this. And then at high cell density, these signals accumulate. Sometimes there's a positive feedback loop, so there's more signals, exponentially more signal that is being produced, but it's not necessary for it to function. And then at some point, these signals that diffuse in and out of the cell will interact with a regulator. And in Pseudomonas aeruginosa is called LASR. And the cognate synthase that makes the signal in Pseudomonas aeruginosa is called LAS-I. Okay? And what this regulator, when it's complexed with signal, initiates is the production of public goods that is now restricted, as I said, to high cell density. Okay? This process can sort of optimize public goods production, but it cannot altogether prevent exploitation, as I will show. And so I'll look at quorum sensing first as a social behavior subject to conflict, and tell you a little bit about how this whole thing started after I got to Oregon State in 2006, 2007. We just thought to do some simple in vitro evolution, so experimental evolution in a test tube, by growing Pseudomonas aeruginosa under conditions that require quorum sensing that haven't been done before, but in a way it is, you know, an infection-relevant condition because quorum sensing is also required for infection in many animal models. Okay, in many respects, this little microcosm does not reflect infection, but this one selection pressure is similar to it. And so we simply grew Pseudomonas in a medium that contained protein as a carbon source. So that requires the LAS-R dependent, that is quorum sensing dependent production of exoproteases. I'm just showing one, but there's a whole pipeline of them that break down protein into small building blocks, dipeptides, individual amino acids that are then taken up by a specific uptake mechanism. So this growth environment is quorum sensing dependent. So you find that a wild type will grow and form this nice characteristic blue-green pigment while doing it, whereas a LAS-R mutant won't. LAS-R mutant just won't grow. So clearly has a fitness disadvantage here. So we kept these guys growing for long periods of time and then scored phenotypes. Skim milk proteolysis was one. So, do these isolates that we obtain day after day, do they still quorum sense? They still produce protease? And we found that after about 8 days, there's an increasing fraction of protease deficient mutants that emerges, and sequencing then revealed that they're all LAS-R mutants. Okay, they are all signal blind. They are no longer able to receive, detect the quorum sensing signal. I did some further experiments, some defined co-culture experiments to show that these are indeed cheaters because they have negative frequency dependent relative fitness. Okay, and defined co-cultures with different initial concentrations of, or initial frequencies of LAS-R mutant. Okay. So these LAS-R mutants emerge, they're social cheaters, and they primarily emerge and evolve under conditions that require quorum sensing. We did also a control, there's just a standard growth medium, and LAS-R mutants don't emerge in that environment. However, we also recorded growth overall, and while this is on log scale, but you might see that there's a slight decrease, there's no population collapse. So the cheaters don't impose a sufficient burden that causes the entire population to collapse. And that is what we might call a tragedy of the commons, which Garrett Hardin coined that term in a 1968 paper, sort of as where population collapse is basically the consequence of the selfish overuse of common resources. And we figured we might test this tragedy of the commons hypothesis in chemostat, which is a continuous culture environment where you would definitely see if a population cannot make it, it would completely wash out, would clear chemostat. Chemostat, just to explain a little bit, it's a bit more technical than your average batch culture where things change a lot. Chemostat, as the name implies, things are supposed to be stable. So you have an inflow port where medium flows in, and then also you have a media outflow where spent medium and bacteria are basically transported out to waste. There's also an air supply. Everything is well mixed. And that results in a situation where you can achieve a steady state environment, okay, of constant cell density, of constant nutrient supply, constant conditions overall, where the growth rate is determined by the dilution rate. And the dilution rate is in turn determined by the pump speed at which medium flows in, okay? And with this system, we either grew the wild type on its own or in a mixture with the LAS-R mutant, okay, with the LAS-R mutant cheater. So we had a wild type only control where we measured cell density over about 200 hours. So this went for several days, this thing. That's the beauty of it, that in principle, you can run these things indefinitely. Initially, there was a batch phase where cells just grew up to the right cell density, and then a flow was initiated, and basically the wild type was able to achieve a stable cell density in this environment. And then we had another condition where about 10% LAS-R mutant cheaters were spiked in, and then of course the outcome was very different. Initially, the growth up to the cell density, to the maximum cell density was the same. But as we initiated flow, all of our replicate populations were essentially washed out within the time frame observed. We looked at the individual replicates a little more carefully, and there slight differences emerged in terms of subpopulation composition. So we looked at the LAS-R mutant, which was conveniently tagged with an antibiotic resistance marker and was easily identifiable. We also measured protease deficiency overall. Replicates 1 and 3 look pretty much where these phenotypes overlap, suggesting that the only protease deficient isolates are indeed LAS-R mutants in that system. But here we had some discrepancies where there were more protease deficient isolates than LAS-R mutants, suggesting that there is de novo evolution of protease deficiency. So there's something else going on, even within this fairly short time frame. But chemostats are known to be highly selective for certain highly adapted phenotypes. And so what we are currently looking at, what are these mutations? Okay, what are these underlying mutations that result in protease deficiency? Okay. Perhaps in a little more applied context, I'd like to say a few things about social interactions and implications for the resistance to antivirulence drugs. Okay. It's probably best introduced by reviewing what antivirulence drugs are and what the difference is to traditional antibiotics. Traditional antibiotics target essential cellular machinery, metabolism, replication, transcription, translation of microbes, and thereby kill them. So resistant strains have a tremendous selection pressure to outgrow this population. Antivirulence drugs work differently. They interfere with specific virulence functions. So they might inhibit a toxin in the host cell. They might inhibit bacterial adhesion to host tissues. They might inhibit toxin delivery or also inhibit virulence regulation. Quorum sensing is one of those. And so in this context, acyl homoserine lactone analogs have been developed. Furanones are one of those types. They function as competitive quorum sensing inhibitors. Okay. So they are virulence drugs. And they're supposed to simply disarm, not kill. So the conventional wisdom or just some preliminary thinking had resulted in this notion that they would not cause resistance, only slowly foster resistance. But of course, resistance should evolve whenever the antivirulence drug inhibits colonization, dissemination, or immune evasion during infection, as was noted in a Plus Pathogens paper. And we wondered, well, what about bacterial social interactions? Would they have an impact on antivirulence drug development? Of course, with particular focus on quorum sensing inhibitors. And the experiment that we did, it was just in vitro. It was not, in an animal model. I would admit was a little convoluted. So I'm just showing the result in a cartoon format. The original paper was published several years ago. So what it was important to consider that there's two different types of behaviors that are distinguished here that are controlled by quorum sensing. Cooperative behaviors, the type we've already discussed, right? Public goods like proteases that are secreted out of the cell. But there's actually also non-cooperative behaviors that are controlled by quorum sensing. Not many, but Pseudomonas aeruginosa has a couple, where, for example, the intracellular utilization of certain carbon sources is, for some reason, also controlled by quorum sensing. We don't really know why yet. But we exploit this fact to distinguish these two types of behaviors in the context of quorum sensing inhibitors. Okay, let's assume that a quorum sensing inhibitor targets a cooperative behavior. And then we have two different cell types. We have the red guy that is a resistant cell that is surrounded by sensitive cells. And then, of course, the red cell would reflect a cooperator that is surrounded by cheaters, and it would be subject to a tremendous cheater burden because it has to constantly give its public goods and help out with the others in the group. So there's a burden that would slow the evolution of resistance. Again, we've only shown that in vitro. Not so with the non-cooperative behavior. Of course, the goods are kept by the resistant cell and is not shared, so it would eventually outgrow the population, just like a regular antibiotic would impose. Okay, so in this scenario, we can say social conflict is an intriguing process that should slow the evolution of resistance when the targeted trait is cooperative. Okay. And for this part of the talk, I'd like to just summarize here that we've established that quorum sensing is a social behavior subject to conflict. And we've identified these LASR mutations that actually pop up in the clinical context all the time. And clinicians have been really puzzled of why they would emerge initially when, why would there be quorum sensing deficiency if quorum sensing is important for infection in the first place? Well, this helps resolve this apparent paradox, although there are other, it's not as easy clinically. Social conflict leads to a tragedy of the commons, but there are mechanisms that stabilize cooperation that can escape a tragedy under the right conditions, population structure, for example, non-social adaptation, and others, as we and others have shown. And quorum sensing and cooperative virulence may also be a suitable antivirulence drug target, okay, as I have just shown. So I'd like to move on and tell you a little more about quorum sensing mechanisms. It's a lot of work that's been done in this area. A lot of labs have contributed to mechanistic sort of picture of this pathway of the LAS-R, LAS-I quorum sensing circuitry in Pseudomonas. So there are other quorum sensing pathways that interface with this, but we'll focus on this one, which is considered to be at the top of the hierarchy, and it influences everything downstream. Okay. So I've already introduced some of the players. We have LAS-I, it's the autoinducer synthase that makes the homoserine lactone molecule that diffuses in and out of the cell, that then binds a LAS-R receptor that dimerizes and leads to a couple of feedbacks, namely signal-bound LAS-R activates LAS-I further and also activates LAS-R in addition to, of course, target genes. Okay, so we have a positive feedback in the system here. Positive autoinduction. There's also a second negative feedback loop that dampens quorum sensing gene expression once it has transitioned to the active state. Okay, and that was important as well, because our next step was to analyze this little circuitry here more formally by mathematical modeling. So we determined, or we formulated a deterministic ordinary differential equation model that had 6 variables: cell density, extra and intracellular signal concentration, and the concentration of these proteins here. And lots of different parameters, and was done in collaboration with Christina Kuttler and her group at the Technical University of Munich. And our goal was with this to identify emergent properties of the system. And our first step was to parameterize the model based on experimental data, basically fit the model to experimental data. Okay, so growth is one simple set of variables that we fit our model to, and so a signal concentration that is easily measurable. We also fit to the concentrations or the expression levels of LAS-I, of RSAL, and of LAS-R by fusing promoters to green fluorescent protein, making the assumption that gene expression is the same as protein expression, which we know may or may not be accurate here, but it's a transcriptional regulatory system. And then we fit the model to the data, and to our surprise, there was an incredibly good fit. So our experimenters worked very hard, and this is something that the modelers rarely see, this nice fit. So we were pleased, and that allowed us to really have a good model to then take the next step to look at emergent properties. So how do these individual components come together to dictate higher level or inform higher level functionalities of the system, such as the long-term behavior or the steady state behavior, right? When conditions don't change in batch cultures, they change all the time. In a chemostat, they don't. So this would be kind of a chemostat-like environment where you keep cell density constant. I mean, you dial in different cell densities, but then keep them growing at that fixed cell density. And then you measure steady state quorum sensing activity. Our readout is the expression of the synthase, which, as I've just shown, is also under quorum sensing regulation. Okay. So, and then what we find is that we really only have 2 states in the system, namely an off state and an on state, and nothing in between. Okay, so there's a very strict separation. So this is a bistable switch. It's quite common in biological systems, not only in biological systems, Earth systems as well. So we have this bistable switch that exhibits hysteresis. So it's dependent on its history. When cells transition from the off state to the on state, there's a certain switching threshold is indicated by this arrow. But if cells are first in the on state and then they switch to the off state, then the cell density of that transition is much lower. Okay, curiously. So you have this window of hysteresis here. And what we are currently doing, or have been up to for the last year or so, is to experimentally test these modeling predictions. Is the system really bistable, and does it exhibit hysteresis? And for that, we devised a sort of quasi-steady state cultivation approach through recursive dilution in batch culture. We had so many conditions to test that it wasn't feasible to go to kind of a clumsy chemostat, which is kind of a low throughput environment. So we use these microtiter plates and the twofold dilutions from a pre-culture to the desired cell densities, incubated for 1 hour, measured cell density and GFP expression, then use that information to dilute back down, to samples back down to the original desired density, grew them for another hour and kept cycling until we deemed that a steady state was reached. And so these are steady state cell density over time. Okay, so you see that cells do cycle. There's a bit of an up and down, a bit of wiggling, but they basically are in their window of, within their narrow range of cell densities over time that don't overlap much. However, when we look at GFP expression, then we find a very different picture. Basically, that cultures either converge to a high density or to a high expression state or to a low expression state of whether they were initially off or were initially on. When we then plot the last time point, the steady state data as a function of cell density, we find this sort of bistable scenario that when cells are initially off, they switch to the on state at a certain cell density. When they're initially on, they switch back at a much lower cell density. So this window of hysteresis really surprised us. It's very wide and is consistent with our modeling predictions in principle. Okay. So this is population level data, right? We've looked at bulk gene expression of thousands of cells, if not more, at the same time. And so what we wanted to also look at bistability at the single cell level using flow cytometry. We had to scale up our approach. It was even a little more cumbersome because we needed to have larger culture volumes that we could subject to flow cytometry analysis. But our approach with reporter fusions was the same, so we could excite individual cells by a laser that would tell us green fluorescent protein expression as a readout. And so informed by our population level data, we chose samples that were really close to the switch points. We had cells that were initially off, or cultures that were initially off. We have a certain unimodally distributed population of cells with a certain median GFP intensity. Okay, they switch in unison, unimodally to the on state here. Cell densities are pretty close together. So it doesn't take much to switch them. Cells that were initially on, they stay on to a much lower density, okay, of the same magnitude, and then they switch all together to the low cell density state, which is only slightly below the on state in that scenario. So that tells us, these unimodal responses tell us that what we have is bistability at the group level, that the entire population switches. And that's what quorum sensing is supposed to do. At least that's the textbook example, right? It causes switching. Everybody's on the same page, communicates to do something together as a population. And that's what we would call group sensing or group level bistability. It's an all or none transition of everybody at a threshold density. But what has been most commonly observed, I would say, in literature is not this. In fact, this has never been observed in a native quorum sensing system, and one engineered in E. coli, but with some caveats. What we see more often is something like this. This great heterogeneity where individual cells switch, but slowly. This causes more gradual induction over time. The mechanism was unclear, but There was an interesting theoretical paper several years ago on PLOS computation biology that suggested that this could also be sort of this gradual transition could be, in fact, cell autonomous by stability, where every cell switches on its own, independent of what the other ones are doing. And that is dependent on the induction threshold. So the induction threshold is the signal concentration at which the cell switches on. If the induction threshold is close to the intracellular signal concentration in the off state, then just stochastic fluctuations might switch individual cells on, but not others. Okay. State switching is generally still dependent on cell density because, of course, the extracellular signal concentration will have an influence about the degree of state switching. It will increase as cell density increases. Okay, so here you have a very different group sensing. It's switch-like in self-sensing. It's switch-like on the individual level, but gradual on the population level. So we figured, how could we investigate this? In our Pseudomonas aeruginosa system, and we focused on the induction threshold and perhaps proteins that can modulate the induction threshold. Namely, there are accessory proteins in the system called anti-activators. I'll tell you in a second what they do. But if anti-activators are present, then you have the wild type situation that we observed. There's a high induction threshold. If you take anti-activators away, so we postulated, you reduce the induction threshold, you might be in a situation where there's spurious self-activation or cell autonomous by stability. Okay, so what are anti-activators? This is again our wiring diagram. The anti-activators come in here. We call them Q proteins because they're called QSLA, QTEE, and QSCR. There are three that have so far been identified in Pseudomonas aeruginosa, so we don't know why it needs three, why one isn't sufficient. Maybe there are even more, we don't know, but they all do kind of similar things. Former graduate student looked at all these anti-activators and their effect on quorum sensing in single, double, triple mutant, all combinations, and found that actually QSLA and QTE, these two had the largest effect, and those were the ones that we focused on. Okay, and looking at it at the single cell level, of the modality of distributions. First of all, in the wild type, we did this just simply in batch culture, not in under steady state conditions now. It's much easier to do. So you find that, there's a fairly rapid transition from the off state here at low cell density to the on state at higher cell density. Again, the entire population switches. As we introduce anti-activator mutations, that picture changes, and we have here in this double mutant, a sizable population that is at about 10 to the fourth, which is fully in the on state, even at a very, very low cell density. This is so dilute that you can't even see the cells. It's kind of difficult to work with them. This is as low as we could get it. And even then, there are cells in the on state, so consistent with our prediction, right? There might be spurious self-activation going on here. So we have the entire sampling range here. We have the wild type, which induces at a much more steeply at a higher cell density than the D of about 1, several orders of magnitude lower, much more sensitive is our anti-activator double mutant. Okay. So what's going on here? Perhaps self-sensing. So we devised a co-culturing scheme that would allow us to test this self-sensing hypothesis. Okay. The two cell types in this co-culture were our focal experimental cell or condition that was LAS-I proficient, so it was able to produce the autoinducer itself. And so in this cell, we would, under certain conditions, observe self-sensing if it happened. And then we had a biosensor of group sensing because it was a LAS-I mutant that did not make its own signal. It required the signal to diffuse out from the producing cell into the non-producing cell and then switch quorum sensing on. So both cell types contained the same transcriptional reporter, the LAS-I GFP fusion, but in order to tell where transcriptional activity came from that we would measure, GFP activity, we labeled them with these cells with a second marker, either DS red or E2 crimson. allowed us to distinguish by flow cytometry. Okay, we pitted them 50-50 in culture, and then predicted the following scenarios. Okay, in the Q plus case, and here we're only considering this double mutant case with a sizable on population. Okay, the Q plus, so it's essentially the wild type background, we would see co-induction of I plus and I minus cells at essentially the same time, because that's what group sensing does. Everybody's on the same page. Okay. In the Q minus background, so anti-activator double mutant background, we would see that an I plus strain, namely this one, an I plus cell, would self-sense, would short circuit, would be constitutively active essentially, whereas our biosensor would not be, because it is reliable on true quorum sensing, on group sensing. Okay. So these are some examples again of flow cytometry data through the growth curve: low density, intermediate density, high cell density. For the Q plus background, the wild type background, what you see is just an almost perfect overlap of the I plus and the I minus. These two panels simply indicate a dye swapping where one is blue and one is red, and then one is red and one is blue. Okay. Now in our Q minus and the anti-activator mutant background, we have this population of the subpopulation of on cells that is only formed by the I plus cells. The I minus cells are still completely off. And this heterogeneity is maintained throughout growth. We have the entire view here. The wild type background, you get perfectly overlapping induction of both cell types, the I plus and the I minus. Not so in the anti-activator deletion background, you have them offset. You have the I minus, the group sensing subpopulation lagging behind significantly. I plus versus I minus can be expressed as a function of cell density in terms of response ratio. So I plus GFP intensity divided by I minus. We'll be doing on time. Okay, all right. And so there's a significant, statistically significant difference in response ratios, with a high response ratio in the Q minus and a response ratio that is close to 1 in the Q plus background. So demonstrating that self-sensing indeed can occur, that a quorum sensing circuitry has the potential to self-sense and short circuit itself. So the conclusions here are that Pseudomonas aeruginosa quorum sensing constitutes a bistable population switch, that it has the capacity for self-sensing, that it's constrained by anti-activator proteins, so that it also ascribes a meaning or a function to these anti-activator proteins. They not only modulate the induction threshold, but they prevent self-sensing. So they're essential for true quorum sensing to work. I think there can be other implications with respect to synthetic biology. You can basically tune the system to the type of induction modality that you want. Do you want group sensing? Do you want self-sensing? Do you want a switch, or do you want graded induction? The type of quorum sensing function has implications for ecology, for bed hedging. This sort of heterogeneity might indicate that diversity might be beneficial, perhaps a division of labor, as it happens in other microbial species as well. Okay. Are we at a good stopping point, or a couple minutes? Then I could, I think I could talk about siderophores, that second topic, and this is just briefly. So as siderophores I've already alluded to are these, iron scavenging molecules that are expressed under iron starvation conditions. And most of them are peptides, non-ribosomal peptides, quite bulky, quite large molecules. This is the one from Pseudomonas aeruginosa. It's called pyoverdin. Okay, it's easy to work with. It has the characteristic under iron starvation conditions. The culture is just sort of bright yellow. It also has fluorescent properties, so it's easy to measure and quantify as well. Okay, it's a tremendous diversity of pyoverdins amongst Pseudomonas species. Interesting from an evolutionary point of view as well. But we look more at the energetics now, because if this behavior, if the production of pyroverdin is costly, then it should, favor cheaters in systems. And cheating has been looked at in the context of pyroverdin cooperation. So we looked at cost that is customary in microbiology from the amount of ATP that is used. And basically wrote up all the reactions in the table and came up with a sum of 26 ATP per pyroverdin molecule. Okay, so it's a non-ribosomal peptide synthesases that put them all in a row, like an assembly line, an accessory reaction. So 26 ATP, that seems costly at face value. So cheaters would spread, so the assumption. But we also considered a more basic physiological aspect, that it should matter what the growth medium looks like, okay, what the resource availability is. And we formulated this hypothesis that fitness costs should be a function of nutrient and public good composition, because these non-ribosomal peptides are largely composed of carbon and nitrogen, right? Because they're made out of amino acids. So carbon and nitrogen, right? So if carbon and nitrogen are limiting, then there should be kind of a bifurcation going on in the cell. Either carbon and nitrogen goes into growth, into biomass, or into pyroverdin production, but not both. So there's a bit of a conflict there. However, when other nutrients are limiting, that is growth rate limiting, such as iron, phosphorus, or sulfur, then by definition, carbon and nitrogen are in surplus. So they can be used at will and be funneled into pyroverdin production. So if we connect fitness cost with propensity for cheating, we could say when carbon and nitrogen limiting, cheaters should invade. And when the other nutrients are limiting, then cheaters should not invade. We first did a flux balance analysis. It's basically a computational model of bacterial metabolism at steady state. And in particular, we added the pyroverdin reactions to an existing model that was already out there. And we looked at an objective function that is biomass production or growth as a function of pyroverdin production. And as you see, there's this trade-off that we predicted when carbon and nitrogen are limiting, up to the point where you produce so much pyroverdin that the cells don't grow at all, right? At this point, the growth rate is essentially zero. But not so with the other nutrients. There was no trade-off at all. For experimental reasons, we didn't go through all, or for, I guess, technical reasons, we didn't go all through the experimental permutations. We just chose carbon and phosphorus as one condition that imposes a trade-off and one that doesn't, and found that indeed in co-culture, cheaters in the form of PBDS mutants, that's a regulator of pyroverdin synthesis, emerge, but under phosphorus starvation conditions, they don't. Okay. So for this part of the talk, it's kind of interesting to see that the fitness costs of secretion are context dependent. Namely, the chemical composition of the public good and the nutrient source can determine the evolutionary stability of cooperation. Okay, that's it. So I'd like to acknowledge the people in my group currently that contributed to some of the work that I've talked about, the 3 grad students, Bryce and Lynn and Anita, and 2 undergrads that I have in the lab, Isla and Jack. Former lab members, so I went quite, a ways back. They're doing other things now, PhD students and master students and collaborators at OSU and beyond, especially the group at the Technical University of Munich that helped with the mathematical stuff. All right, thank you very much. Yes, please. Mm-hmm.
Just what about what siderophore how far away from the cell do these siderophores typically diffuse within the time scale that it matters, versus what is the typical cell-to-cell distance in your cultures? And do you think that the ratio of these two distances also affects the propensity for cheating?
Yes, definitely. This is spatial structure doesn't matter, or population viscosity. So cooperation is favored, as has been shown, if cultures are grown on plates, simply on plates, and there's certainly diffusion is limited. The cells stick together in colonies. That's a very good point. And under those conditions, you find little, if any, cheating. Now, these conditions here were a well-stirred, mixed environment. It was a chemostat environment as well, but well-stirred. So you basically have a homogeneous distribution of cells as well as siderophores in that system. So of course, we chose a system where cheating would be, favored. Yes. That's a good point. Yeah, so spatial structure is a great, and also seeding dispersal. As a colony breaks up and seeds new territory, then when there's going through bottlenecks every time, there's also just stochastically a chance that cooperators will be on their own, cheaters will be on their own, which then would be disfavored. They won't grow at all. So yeah, so just the life cycle can also help. Yes.
Might be that you still make the siderophore, but the adapter was totally take out more of it.
Yes, that's a great point. That's also something that we've looked at and is known as that different Pseudomonas compete or pirate siderophores. We've worked with another species, Pseudomonas protegens, which is cell bacterium, a rhizosphere bacterium that's also used for biocontrol, actually. And it has a tremendous number of pyoverdin receptors. So it has the ability to take up pyoverdins that are produced, and other siderophores actually, that are produced by other species. It still makes its own pyoverdin, and it has the ability to dial its own expression depending on what else is available. And it will carefully fine-tune. Initially, we were surprised. We gave Pseudomonas fluorescens siderophores from Pseudomonas aeruginosa that it can readily use, but it still made its own siderophores. But then careful measurements just revealed a small difference that they dial back their own production, but not by much, because self-pyoverdin production also gives a competitive advantage by depriving competitors of iron. So there's this effect as well. Yeah. Yes, please.
I'm interested in the difference between the really strong biostable population level versus that more fragile. And so it's curious about whether it's characterizable natural variation and whether we're seeing these two different patterns associated with your hypothesis about related to that. There is natural variation in different species of bacteria in different contexts, and these two behaviors. Is that associated with things like more or less environmental variability, and that's just a benefit from having strategies? So do we know what the differences?
Those would be great, great questions to explore, to write into a nice proposal or something. No, those are things that have not been looked at. I mean, I also don't. I mean, there are pathogens where this sort of this self-sensing behavior is seen, just trying to sort it based on environmental, niche or habitat. There are there's rhizobia, where this has been observed as well. So I can't really answer that with great confidence, whether there's an environmental effect or whether there might be a benefit to this diversity in terms of bed hedging in one environment versus another. Yeah. Yes.
How do you person if there was anti-activated proteins? Is anything there about evolution of those?
Also, a good point. We are not into sort of genome analysis. We've looked into this a little bit, and all of Pseudomonas aeruginosa has these essentially, and as all three of them, even though some strains have, for example, one of the anti-activators called QSLA is mutated, is inactive in some strains. And hence, those strains are actually hypervirulent because you take away quorum sensing, to take away an inhibitor of quorum sensing. So these genes are expressed more highly. There are completely different anti-activators that have evolved independently in agrobacteria, for example. There's a quorum sensing system that's called TRA that helps with the transfer of the plasmid that induces tumors in plants. And there's an anti-activator called TRA-M. There's no similarity to any of the three. So it's likely that this mechanism evolved independently and yet others are likely to be discovered. Yeah. Good point. Yeah, thank you.